Hello and welcome to our webinar Wednesday lineup. I am Helmut Brechel, Product Manager for Molecular Biology at Biosym. We are happy that so many people, customers and curious scientific workers join our webinar today. Thank you very much. The aim of Biosym webinars is to transport new and exciting information about laboratory technologies and new products to all interested people. Today, we would like to present an instrument line for microliter spectroscopy and fluorometry, the Denovix DS11 line. And we are pleased that Andrew Jones from Denovix will introduce the exciting devices and technologies. Andrew is Market Development Manager at Denovix with much more than a decade of experience in microliter spectroscopy. At the end of the session, we will have a few minutes for discussion. Please type your questions via chat into the system whenever you like. We will try to answer them at the end. Thanks in advance for your input. Andrew, now please give us an insight into the DS11 line.
Hi Helmut, and thanks everybody for joining us. As Helmut said, I'm the Market Development Manager with Denovix. I'm based in the UK. So today I'm going to take you through the DS11 series of spectrophotometers and fluorometers. And the way we'll do this is I've got a couple of slides I'll show you. So not too many, just a few slides. And then I'm going to switch to a hands-on demo. So I'm going to take you through some of the key features and the benefits of the DS11 through running some samples, running some DNA samples through Absorbance, and also introducing the fluorescence capability of the instrument. So a little bit about Denovix to start. Denovix, if you're not familiar with us already, Denovix was founded in 2012, and it was set up initially by Fred Kielhorn. And Fred Kielhorn was one of the original founders of nanodrop technologies alongside his three partners at the time. We're based in Wilmington in Delaware in the US, and all of our operations, apart from me and my role in the UK, actually take place in Wilmington. So we have all of our manufacturing, all of the product support, all of the software development, all happens in a single facility, and we think that's important in terms of developing products that that reflect what life scientists want by having applications teams working very closely with our development teams. So Denovix are the inventors of the multi-award-winning DS11 series that we're going to see today, and the new cell drop automated cell counters that allow you to count cells without slides. If you're interested in those, we also have a webinar available through Biosym on that subject. And one of our focuses is really about having the best in our industry in terms of technical support and support for our customers and for our distributors such as Biosym. And recently we were awarded the Reviewer's Choice Life Science Customer Service Company of the Year for 2020, which we were very proud of. You can see the badge at the bottom of the screen there, along with numerous other awards that we've been really pleased to accept over the last few years for the DS11 series and for cell drop. So moving on to the instrument itself, this is the DS11 series that you see on the screen at the moment. And as part of this instrument, we have the choice of up to 3 different measurement modes. So we have microvolume absorbance, we have UV-Vis in cuvettes, so Cuvette absorbance, and we also have the option for an integrated fluorometer. And this is a 4-channel fluorometer. The instrument is fully connected to your network, so we can connect through Wi-Fi and through Ethernet. And this allows us to connect to network folders, to printers, to handle data through email. And it also has 3 USB ports that allow you to connect things such as label writers, barcode readers. We can connect USB memory drives, or if you so want, you can connect keyboards, mice, that sort of thing. So I mentioned we have 3 different measurement modes, and you can choose between these measurement modes in any combination. So we have our microvolume, our cuvette, and our fluorescence, and this slide just shows you that for any selection of those different measurement modes, we have a model available for you. So you can basically select an instrument based on your current needs in your lab or what you project to be your future needs. Response to our instruments from our customers. And the DS11 series itself has had over 300 independent customer reviews posted online to sites such as Select Science. And I just brought up a few of the reviews here just to show you an example. And people really talk about the ease of use, the reproducibility, the reliability of the instrument, and the flexibility of the assays and the measurements that we can perform on the instrument. And for any of our anyone who's looking at the instrument or has a need for an instrument like this, we always encourage people to go online and really take a good look at the customer reviews and just see the experience of other scientists who've been using the instrument for years. So one of the key differences and one of the key innovations brought to the market by Denovix was the introduction of our patented SmartPath technology. And SmartPath technology was really introduced and developed to solve one of the common issues with microvolume spectrophotometers at the time in the market. To give you an idea of what SmartPath does, is essentially SmartPath is a combination of software algorithms developed by Denovix and hardware integrated into the instrument that allows us to get real-time feedback on the absorbance as we're measuring. And as we measure, we get this real-time feedback on the absorbance, and we're able to shorten the path length in order to hit the optimum path length for every single sample that we measure. So as we'll see when we go through the live demo, the sample is always compressed and it's never stretched, and it's compressed to the optimum path length. And the important thing about compressing the sample is that this solves one of the issues that I mentioned earlier. This solves the issue of broken sample columns. And you find that with all the instruments out there that come down, touch your sample, and then stretch your sample into a column, you'll find that this has the potential to break the column of the sample. And as soon as that column is broken, it leads to erratic results and unreliable results. Now, this is especially true in protein labs, where your sample may well be in low surface tension buffers. And so very often when people who are attempting to measure proteins look at microvolume and they think that microvolume doesn't work for protein measurements, that's not true. The DS11 series works excellently for protein measurements. It's just understanding that some microvolume spectrophotometers won't work well for proteins because they stretch the sample and the sample column breaks. So SmartPath allows us to hit the optimum path length to improve sample reproducibility and accuracy. It ensures that there's no column breakages. So again, improving the accuracy of our results. And a third advantage of SmartPath technology is that the instrument is always calibrated. So every time we start up a measurement app, it will go through a initialization routine. And part of that initialization routine is to find its home position, and it always knows how far it has to move to get to any path length. So it doesn't require any additional calibration or additional calibration checks that many of our competitors would do. Clearly, the advantage of that is that you don't have drift over time, and you can have total confidence in your measurements. As I mentioned, we also have fluorescence as an option on the DS11 series, and I'm going to take you through some fluorescence measurements today during the course of the presentation. And the advantage of using fluorescence in addition to absorbance is fluorescence gives you a very sensitive and specific quantification. So it's sensitive in that it will, for example, for double-stranded DNA, allow you to measure down into picogram per microliter concentrations. So with our kits that I'll talk about in a little bit, we can measure down to 0.5 picograms per microliter. And it's specific in that all the kits are specific for a particular type of analyte. So for example, you would get a kit that is specific for double-stranded DNA, or a kit that is specific for RNA. So it won't quantify and it won't be affected by any other nucleic acid species in that particular sample. So fluorescence will improve our lower detection limit. It'll give us improved sensitivity and reproducibility. And our fluorescence instrument that we have integrated in this system is particularly flexible compared to any competitor instruments, standalone fluorometers, because we have a 4-channel system compared to 2 in most instruments. So we have a UV, a blue, a green, and a red light source. And what this allows us to do, of course, is the more light sources you have of this nature, the greater the variety of fluorophores and the greater the flexibility of the system to run additional assays is. And I'll show you as we go through the course of the presentation how we have Denovix assays pre-programmed into the instrument, but also a number of common fluorophores and common assay kits that people might already use pre-programmed for user convenience. And I mentioned that for fluorescence, Denovix do have a range of assays available. And the advantage of the Denovix assays compared to our competitors is that all of our assays have a far wider dynamic range than our competitors. So, for example, we can see our double-stranded DNA assays at the top here, and you can see compared to our or compared to one of the bigger competitors that we have, each of our assays, the broad range, the comparative broad range, high sensitivity kits have a greater dynamic range, and we also have an ultra high sensitivity kit, which there is no comparison to in our competitors. So we can go down to 0.5 picograms per microliter on that. And a similar situation for our RNA assays, where we have a single assay that replaces 2 assays from our competitor. Advantage of that clearly is its simplicity of ordering, but also we have a greater dynamic range, so more samples will fit into the dynamic range of that assay, and you don't have to go and rerun samples or dilute samples in order to get them into the range of the assay. So that's just a little introduction to the kind of key technology around the DS11 series. What I'm going to do now is I'm going to switch over to our live feed. And should be able to see the DS11 there now. Great. Okay, so the instrument I'm going to show you is the DS11 FX Plus instrument. And this is the instrument that combines all 3 of our measurement modes that I discussed earlier. So if we go to top here, what we'll see, if we look at the top, at the front of the instrument, we have a microvolume platform. So our pipette, we'll pipette 1 microliter of our sample onto the front pedestal here. And then we move the arm down and it will take a measurement. So this is microvolume. To the rear, we have a heated cuvette place. So we can heat 37 through to 42 degrees Celsius in the cuvette station. And that's full UV-Vis as well. And so people generally use that for things like biochemical assays, kinetic assays, or quite often people will use that for OD600 measurements, so for quantifying microbial cell cultures. That takes all standard cuvette types, So whether they're quartz or disposable, they can fit in there, and they come with a variety of different path lengths, and we have all of those hardwired into the software, so you don't have to do any calculations yourself. To the right of the instrument, we have our fluorescence port, and for fluorescence, we use the 0.5 mil thin wall PCR tubes, and I'm going to take you through some fluorescent measurements during the course of the presentation. The other thing you'll see on the key plate at the top is in this recess here, we have a high precision screw. And it's the high precision screw that works with the software algorithms to set the height of the sample path, so the path length of the instrument. So that's a key component for us for smart path technology that I mentioned earlier. If we come back to the front of the instrument, we'll zoom in a little bit. You'll see that it's all powered by a high definition touchscreen interface. And you'll notice that all of our measurement applications are software apps. So along the top, we have a number of square apps. And again, on the bottom, a number of square apps here. All of the square ones are absorbance. And so we separate those out into double-stranded DNA, RNA, single-stranded DNA, protein, and some other applications that we can go through during the course of this. And the reason for separating those out is really just to avoid any errors. So we color code all of our apps, we label them, and we separate them out so that people aren't mistakenly going into an RNA app and measuring their DNA sample. Similarly color-coded to our absorbance apps, and again, separated out into different analytes. So again, it's just a way of ensuring that it's a very easy to use, simple system. We can swipe on the screen and it takes us to our second screen where there are an additional couple of apps here for peptides and kinetics. And we also have some housekeeping options and settings on this page. So we have the ability to go and check the system is working with all the published specifications using the diagnostics apps. So although we don't require any calibration, you can check that everything is working according to the specifications using these apps. We have a calculator app that allows us to go in and do dilution calculations and various other things in there. We can set up user accounts, and these are password protected user accounts. So you can set up the instrument as you like it and set up default values specific to you. Timers, general settings, updaters for software, and all of our software updates are free. And also we can set this instrument up with an email address, and I'll show you why that's a really cool feature as we go through measuring some samples. So first thing we're going to do is we're going to load a sample. So I'm going to, during the course of this presentation, I'm really going to focus on double-stranded DNA, but everything that I show you is really applicable across multiple different analytes. So for DNA, RNA, single-stranded DNA, protein, they're all very similar, but for convenience, we'll use double-stranded DNA. And so we'll just come into the software, go through a quick initialization. And the first thing it's going to ask us to do is to put on a blank measurement. So you'll notice that when we take a look at the screen here, you'll notice that the screen that we have here has blank and measure on the top on the left-hand side. And until we've provided a blank, the measure button will be grayed out. So we're going to put on a blank measurement. And the blanks are always important because the blank should be basically whatever your sample is suspended in minus the analyte that you're trying to measure. And we'll just move that up a little bit. And so what that does is, if you have the correct blank on there, it's really important because it allows you to actually look at the quality of your sample as well as just the quantity. So we're going to pipette 1 microliter onto the sample pedestal, put the arm down, and we're just going to hit blank. And so what that'll do is it'll go through the various path lengths, it'll take reference spectrum, and it's going to take that away from our sample when we measure our sample to ensure that we're actually getting an accurate measurement for specifically what our sample is, and not the blank, not the solution that it's loaded in. Just takes a couple of seconds, and when it's done, all we do is we wipe the sample surface, top and bottom, and we're ready to measure our samples. So I'm going to put another sample in. Let me just prepare that on. So at this stage, if we want to, we can put in a sample name. So we'll call that ABC. And I'm just going to put my sample onto the measurement surface. The light flashing and the arm moving down, and you'll see that already that's taken the measurement. We're going to go. It's taken, and then we just wipe off our sample as we did with our blank with just a dry laboratory wipe. That's all is needed. So taking a look around the screen here, you'll see that as soon as we've measured the sample, a trace appears here, and we can zoom into that trace. We can take a look, see if there's any strange peaks or anything. So a kind of first look at if there's any quality control issues with our sample. And there's little buttons here for zoom in and zoom out, or you can pinch and zoom. On the right-hand side, you'll see that we have our concentration. So we have a concentration here in nanograms per microliter. We also have the absorbance at 260 nanometers. We'll just come in a little bit closer. And we have 2 purity ratios reported. So the 260-230 and the 260-280 ratio. So these allow us to get a really good look at, is there protein contamination in our sample, or is there have salts been carried over from our DNA extraction using the 260-230. Now, one of the things that the DS11 has is that it has built-in thresholds for the purity ratios, and it alerts users to any samples that are potentially contaminated. Okay, so basically you don't have to do anything. And what I'm going to do is I'm going to measure another sample that hopefully isn't as good as the first sample. I'll just pipette that on. And actually, I'll show you an additional feature that we have here, which is auto run. So you'll notice that this auto run box has gone yellow, and that just tells us that the arm is up. So there's a sensor in the arm. And if we click that box on, as soon as we drop the arm down, it will measure the next sample. So we don't have to click measure for each of our samples. So that's measured our sample. We'll just wipe that clean, and I'll disarm auto run for the time being. And if you look at this sample, you'll see that the data is similar in many ways. It looks like a great trace still looks fine. Looks like a good sample. We've got a concentration value and our absorbance. But you'll notice next to our 260-230 ratio, we now have a yellow triangle. And the yellow triangle is our contamination alert. So if we click on the yellow triangle, it will show us that there's potentially a problem with our sample, and it will give us a list of things that it could be. But in addition to it being a problem with your sample, it could be something methodological. So it also suggests additional things that you should check. Things like, did you get the blank correct? Was there a sample dried onto the surface when you put your blank on in the first place? Okay, so it offers some procedural things to check, as well as potential sample issues. And it will do this for 260, 230 and the 260, 280 ratio. So you get all of this guidance about what could be the issue with your samples as we go through. We're clearly measuring in microvolume at the moment, but if we do want to measure in cuvettes, it's a simple drop-down menu. And as I mentioned earlier, there are different cuvettes in here with different path lengths. So if you want to use smaller volume cuvettes, we can accommodate those, and all the calculations will be done automatically for you. So at the moment, we've got two samples in our buffer here. Now, it's important to mention that as soon as you measure any samples on the DS11, as soon as that data is collected, it's saved to the instrument. So we have a large onboard memory where every sample is automatically saved, and the system can save something in the region of like 30 million samples. So it's not something that you fill up with data. Okay, so it's not something you have to regularly empty or anything like that. But I'm going to show you how this data is handled and what our reporting options are. So we've run a couple of samples. It's also for us. Automatically for us, and this is kind of a summary of our data. So we've got our sample names, we've got the concentration, we've got the ratios, and the date and the time that it was taken. You'll notice that our alert is also carried over onto this screen. So if there's any problems with any of our samples, we don't lose that data after the run screen. That's carried through into our archive and into our reporting. At this stage, we might want to export our data. So we just go into this share button in the top right-hand corner. So we select the samples, hit share. And this is where we can select from a variety of ways of handling our data. As I mentioned earlier, we have the option to email our data. So we can set this instrument up on our network, and we can set it up with an email address. So it can be as simple as we collect our data, come into this screen, select our email address, and hit OK, and it will email our data to us. And so the data it emails to us is a .csv file. And that .csv you can open up in Excel or a similar sort of editor of that nature, or it's compatible with LIMS systems, for example. And that has all of the raw data in it. So it has all of the data that allows you to regraph your sample, as well as the summary data that we saw in the report table on the previous screen. So we can email our data. We can set up shared folders on the network. So if we want to save our data into a shared folder, we can do that. We can save it to USB, to a label printer, or we can go and print it to a network printer. And I'll show you the network printing in the next screen. So this is the report tab. We also have the option to overlay it graphically. So here's our data overlaid graphically on top of one another. And as before, we could share that data as well. And if we go basically what will happen is it will go to your network, find the, we're not on the network at the moment, but it will go to the network and it will find your network printers, and it will give you the option to print a report like this. So you will get the overlaid data with the table of information underneath. So you have lots of different ways of handling your data with the DS11 series. We can either do it through a table or graphically. And if we want to, in the report screen, if we want to add sample names and edit sample names here, we can do that rather than adding them on the initial run screen. So if we don't want to do it at the same time as running our samples, we can come in and we can edit our sample names at this point. So at this stage, I'm just going to come out of double-stranded DNA and I go back to the home screen. And moving through the apps, RNA and single-stranded DNA are very similar to the double-stranded DNA application. Let's just come in a little bit here. Protein is slightly different. We measure proteins at 280 nanometers rather than 260 as we do with nucleic acids. The look and the feel of all of our apps are very, very similar. And what that means is that, when you've worked through one of the applications, it's very simple to go and work through the rest of them. The difference with the protein measuring at A280 is that proteins are much more diverse in the degree to which they absorb the light. So they all have their own extinction coefficients. Now, we have a number of standard proteins built into the software. And we also have the option to put in your extinction coefficients, your molecular weights. But you don't need to do that for every sample every time. If you use a particular range of proteins, you can add that to the list in here. So you can just use the little 3 squares next to the drop-down menu, and you can define your own extinction coefficient for your protein and save it to this list. And you can do that through for everybody as a user or just in your own individual user account. Everything else is very similar. All the exporting and reporting is exactly the same. So protein at 280 tends to be used for purified proteins. We also have the option to do full UV-Vis analysis. We can do colorimetrics such as Bradford assays and BCA assays. And just coming down to sort of round out some of the absorbance ones at the bottom, if we have proteins or DNA that is labeled with a fluorophore and we want to quantify that using absorbance, we can do that using these two measurement apps at the bottom. We can quantify microbial cell cultures using the OD600 app. And if we don't have a specific app for your application, then you have the option of 2 different custom apps here. And the custom apps allow you to either create standard curve methods, or it allows you to define a measurement range and apply a number of formulas to any absorbance values at particular wavelengths using the formula methods. So in the course of this presentation, I'm not going to go into too much depth in terms of those apps, but we are available or the instruments are available for trial, and we're available to take you through any of these in more depth if you need them. So I'm going to take you through a little bit of fluorescence, and we're going to go into the fluoro double-stranded DNA app to start. Actually, we'll just come out of this and go into it fresh so that you can see how it looks from the start. So as I mentioned earlier, the advantages of fluorescence tend to be that it is more sensitive than using absorbance, and it's very specific. So if you have a mixture of DNA and RNA or fragmented DNA in a sample, it will all absorb it. With fluorescence, it's specific for a particular analyte. So you'll be specifically measuring DNA, for double-stranded DNA, for example. The downside of that, or the advantage perhaps of absorbance, is that for fluorescence, you don't get any information on contamination. And for this reason, we think it's a great idea to be combining absorbance and fluorescence measurements to get very specific measurements, very sensitive measurements, but also then been able to use Absorbance to get contamination information. So coming to the app, you'll notice it's very similar in design to the Absorbance apps. The first thing we want to do is select an assay, and we do that from this drop-down menu. And hopefully you can see here that we have a number of assays. Denovix assays, but also things like quantity assays, Cuvette assays, quantifloor assays. So all the common assays that people use out there are all pre-programmed, just to demonstrate that the system can run all of those assays and you don't have to worry about setting them up yourself. So we're going to use the broad range assay from Denovix, and then we just say we want to use our normal standard of 2 pre-configured standards, and we'll generate a new standard curve so I can show you just how quick it is to measure. So it's going to come out a little bit here. So with fluorescence, first thing we're going to do is it's going to ask us for the blank or the kind of 0 point on our standard curve. I just put that in and I hit measure. And that takes about 2 seconds to measure. That's just done. And then it's going to ask us to put in our top standard. So it's only a 2-point standard curve, which is a blank and a top standard because the assay is linear. And we just hit measure for that. And you'll see within a couple of seconds, we have generated our standard curve. And we're ready to run our samples. We can put replicates in there if we want, but it's generally not required. The reproducibility of this assay is so high that we generally don't need any replicates. So we'll just switch to samples. And we can put our sample name in if we want to here. I'm going to skip that for this particular assay. We'll put our first sample in. Hit measure, and again, what that's going to do is it's going to take the fluorescence measurement, it's going to compare that against our standard curve, and on the right hand side it gives us our concentration. So it's given us let's go a little bit. So it gives us our concentration against the curve. You can see that on the left hand side the little dot has appeared on the curve. This is where it's measured, and just to show you how quick it is to go through and measure a number of samples, we can go and measure a second sample here. And you'll see within a couple of seconds, we've done a standard curve and 2 samples, and we're ready to go. So for this second one, you can see our concentration is 236. With any of these assays, you have the option to expand the assay by adding a different volume of sample. So you can add between 1 and 20 microliters of sample. And this box allows you to define how much you've added. So we're always adding 10 microliters of our standard, but if we add 1 microliter, for example, of our sample, you can see it does a calculation for you at the bottom, and it tells you actually what was the concentration in your original stock concentration, in your stock tube. So fluorescence is very straightforward, very easy to do. The assays are just mix and measure assays. They don't take long to put together at all. You don't have to kind of incubate them for a long time. They're just ready to go. All of the reporting is the same as we saw for Absorbance. And also we can overlay things in a graphical way, and we can print that in the same way as we did for Absorbance as well. So fluorescence is a really nice way of getting specific and sensitive data on our sample. And we can do that for DNA, as we've just seen, but we also have RNA assays, single-stranded DNA, and protein applications on here as well. If you want to use it for kind of basic fluorometry, we also have a Fluorometer module where you can just go in and choose the particular wave or particular light sources that you want to expose your fluorophore to, and just get relative fluorescence values for those. earlier, all of our data is saved to the instrument, and we use the data app to access this information. So the data app is really our window on our database on board, and it allows us to search by application. So you can see we have all of our measurement applications in the drop-down menu. We'll select double-stranded DNA. We can choose a sample name. So if we specifically want to search for a sample, we can do that on sample name or within a date range from when the sample was measured. We'll hit search, and you'll see that it just takes a couple of seconds to, or less than a second, to search the entire instrument for all of this data. And we can come in and we can select data that we've captured today and compare it with data that we measured over various days here, various measurements sessions. We'll add that to the report, and you can see it's now generated a report with all of that data from over the course of, well, this is over the course of many different years, so over the course of about 3 years of this instrument's life. And we can look at those graphically, and we can choose to view any of those data points on top of one another. So it's a great way of seeing how, for example, samples perform over time, how assays are performing, seeing if assay reagents are dropping out, or just putting data together for a presentation or for a publication. And all of that is, again, exportable through email, network, USB, or label or network printers. So I think as we are coming to the end of our allotted time at the moment, I'll leave it there. So in summary, the DS11 series is a multi-mode instrument that combines absorbance in microvolume, in Cuvette, and in fluorescence. And it's unique in the market in being able to do that. The advantage of being able to combine those, as I've discussed during the presentation, is that you're able to get total sample quality control. So you can use with fluorescence measurements that will give you specific measurements for the quantification of that particular analyte. The software itself is very easy to use. You've seen during the course of the demonstration that the apps are very similar. The data is very clearly laid out and very easy to use. So once you know one app, you can comfortably use any of the other apps. And the instrument itself doesn't require any additional calibration. It's very robust and very reproducible and very reliable. So we'd encourage you to ask any questions that you have in the chat window, and also we're happy to follow up with you on any requests for further information on any of the apps or any additional questions. But I think I'll hand over to Helmut and see if he has any questions for us.
Thank you very much, Andrew, for this nice and interesting presentation. So I want to admit that in Germany and Austria, you can contact Biosym for further information or quotes. And if you are interested to working with DS11 or also with the fluorescence unit QFX in your lab, please just contact us directly or local sales account managers for a demo. Yeah, and about questions, I think we can start with, when should I use A280 or colorimetrics? It's protein measurement, I think.
Yes. Yeah. Okay. Yeah, so with the A280 module is really designed for purified proteins. So I think purified proteins measured at 280 nanometers. If you have things like cell lysates where you want a total protein measurement, or if you have buffers that might interfere with the measurement of at 280, then we would recommend using either a colorimetric assay such as like Bradford or BCA or the Lowry assay, which are indirect protein sample against known concentration proteins. So generally, when you have things like Ripper buffers, which absorb very highly at 280, it's a good idea to use a colorimetric, because it's difficult to distinguish your protein from a very highly absorbing buffer. You also have the option to use protein in the, use fluorescence protein in those examples.
Mm-hmm. Next question is a DS11 protected against computer viruses?
Ah, right. Yes. So, so the DS11 series is built on a custom Android platform, and the whole environment is what they call sandboxed. So nothing can be installed on the instrument that isn't specifically signed and authorized by Denovix. So, so when it comes to talking to IT departments and getting this implemented on networks, it's a very secure environment because it doesn't allow any additional software or malware to be installed on it. And I guess I would say that over the course of 6 years or so, in thousands and thousands of labs, there's never once been an example where any sort of malware has been installed on a DS11 series. It's a very secure system for putting onto networks.
One short question, maybe you have already covered that. What maintenance recalibration is required?
So yeah, I touched on one of the advantages of SmartPath is that it doesn't require any additional calibration. So everything is calibrated when it leaves the manufacturing plant in Delaware. When it comes into your lab, there really isn't any setup or an initial kind of calibration that you have to do, and there's no periodic maintenance that you need to do. So it will always be calibrated, and the only thing that you should do really is put good practice in place in your lab to make sure that people are wiping it clean between samples and leaving it clean. There is actually one message which didn't come up here because you have to leave it for a few minutes, but it does actually detect. This instrument detects if the arm has been raised between samples or not. So if somebody leaves a sample dried onto the pedestal, it will actually warn you that the arm hasn't been raised, so the sample hasn't been cleaned, and you can then go ahead and clean it. But really, the only maintenance that's required is it's just wiping it clean after you use it. Hopefully that answers the question. Okay.
Okay. So. Again, thank you very much, Andrew. Our time is over, unfortunately. Other questions raised in the meantime will be answered separately by email. And I can say thank you very much for attending the webinar. Have a nice day and goodbye.